I thought it was worth introducing this video by saying that even though this video has been made using the AQA specification as its guidelines, having looked at the OCR specification carefully and exam questions, the content of this video is going to provide you with 99% of the information that you need for nearly any A-level syllabus. And I have shown that by the exam questions that I've looked at the end, which cover a variety of exam boards. Enjoy.
This might be a little bit of a marathon session, but I'm hoping to get through the whole of AQA respiration in this video. Glycolysis, link reaction, Krebs cycle, and the electron transport chain. So just a quick bit of context. But before that, let me just point out, I've got the AQA specification up on the screen so that you can have a look through exactly what you do need to know. But this video is also going to be going through things in the light of the exam answers. So I'm going to be talking to you about AQA respiration in terms of the phrases and terminology that are used in exam answers. So quick bit of context, respiration occurs so that ATP can be produced. It has to happen in all cells all of the time. Notice that respiration doesn't really happen in organisms, it happens in cells. It's a chemical reaction. And the raison d'etre for the chemical reaction is to produce ATP as a form of chemical energy, not to produce the products of the reaction, which are carbon dioxide and water. That, to be quite honest, is neither here nor there. Carbon dioxide is poisonous, and water can generally be got from other places other than a reaction taking place. So now let's start with respiration and where it happens. So I'm kind of hoping that you might recognise what I've drawn here. It is part of a mitochondria, and this is obviously in a eukaryotic cell because only eukaryotic cells have mitochondria. So that means that all plant cells, all animal cells, all fungal cells have mitochondria in them. Prokaryotic cells don't have mitochondria, and they carry out the part of respiration that needs to happen in mitochondria actually across their cell surface membrane. But don't worry, you don't need to know that for your A-level. So let's start by giving you a quick summary of where you were at GCSE. And I think you might find it easy to split up where each of these parts of the reaction occur in respiration. This is a very crude summary. Don't take it too seriously, but you might find this helpful. So, glucose is broken down into carbon dioxide primarily in glycolysis and in the Krebs cycle. Whereas oxygen is formed into water in the electron, or as part of the electron transport chain, the last part of respiration, or the last part of aerobic respiration, because I've deliberately coloured Krebs and the electron transport chain in blue, as this only occurs in aerobic respiration. Whereas glycolysis, that's in purple, and that occurs in anaerobic respiration. The other thing that I just wanted to do was to draw my little tiny summary of aerobic respiration, and this is what I sometimes scrawl in the margins of an exam paper so that I can just keep my head around each part of respiration. So glucose gets turned into 2 pyruvates. And that is the anaerobic part of respiration. And then 2 little lines here for the link reaction, and then we've got 2 Krebs cycle. Note I don't label anything, and then all of the coenzymes, the NADH and the FADH2, is funneled into the electron transport chain. And once again, the blue is denoting the aerobic part of respiration, and the purple is denoting the anaerobic part. Now let's get down to doing some quick labeling of this diagram. I'm hoping that you are recognizing that outside here is the cytoplasm. And this is the outer mitochondrial membrane. This one is the inner mitochondrial membrane. And funnily enough, this space in between them is the intermembrane space. And these projections going into the mitochondria are cristae. A single one is called a crista, but if there's 2 or more than 2, they're cristae with an extra E on the end. And this internal gel-like filling of the mitochondria is the matrix of the mitochondria. And now I think we're ready to start the whole process of photosynthesis. So let's start with glycolysis. And I'm writing this here in the cytoplasm because that is where it takes place. And we start with glucose, which is a 6-carbon compound. And this is phosphorylated by ATP to produce glucose phosphate, which is also a 6-carbon compound. And it is literally the glucose with 2 phosphate groups hanging off each end of it. And that requires 2 ATP because it needs the 2 phosphates from the ATP. The phosphorylation of the glucose actually makes it a lot more reactive. And what happens is that it then splits into 2 3-carbon sugars, which are triose phosphate. Triose does what it says on the tin. Tri means 3, ose means it's a sugar. And then each of these triose phosphates are then oxidized into pyruvate. And one way that we know that they are oxidized is because during this reaction from triose phosphate to pyruvate, NAD is reduced to NADH. And for each of these reactions, 2 ATP is released. So during glycolysis, we put 2 ATP into the whole process and we get 4 ATP out. And therefore, there is a net balance of 2 ATP from glycolysis, as well as 2 molecules of NADH. This is the end of the anaerobic part of respiration. In animals, the pyruvate is turned into lactic acid, and that is so that the NADH can be recycled back to NAD. And in plants and in microorganisms, the pyruvate is turned into ethanol and carbon dioxide. And once again, the sole reason for that is so that the NADH can be recycled back to NAD. This recycling of the NAD/NADH during anaerobic respiration allows anaerobic respiration to keep going, and therefore is crucially important. So we're now switching colours because all of the following processes occur during aerobic respiration only. And this is started off by the pyruvate being actively transported into the mitochondria. The mitochondria can only use pyruvate as a respiratory substrate. It can't move glucose into the mitochondria and it can't use glucose. It can only use pyruvate. So now we're getting to the link reaction. And this is where pyruvate is turned into acetyl-CoA. Pyruvate is a 3-carbon molecule and acetyl-CoA is a 2-carbon molecule. So very logically, it means that a carbon needs to be lost. And when a carbon is lost, it is always done in the form of carbon dioxide. So here we can see pyruvate being changed into acetyl-CoA, losing a carbon dioxide so that it goes from 3C to 2C. But that isn't the end of the story. And what you do need to recognise is that pyruvate reacts with coenzyme A to form acetyl-CoA. And this also is an oxidation reaction because once again, NAD is reduced to NADH. So you could describe this whole reaction is that pyruvate reacts with coenzyme A to form acetyl-CoA and carbon dioxide, and it is an oxidation reaction and it causes the reduction of NAD to NADH. It is a redox reaction. So now we're about to move into the Krebs cycle, and acetyl-CoA is fed into the Krebs cycle and it reacts with a 4-carbon compound to make a 6-carbon compound. And the coenzyme A is actually recycled back for it to be used again. You don't need to know any of the names of the intermediates in the Krebs cycle, which is why I'm only referring them to 4C and 6C. For those of you who are going on to do something biological at university, it may be useful for you to know that the 4-carbon compound is oxaloacetate and the 6-carbon compound is citrate or citric acid. And the reason why it's useful to know that is that as well as being known as the Krebs cycle, this cycle is also known as the citric acid cycle. Now I'm going to add in just a little bit more detail to this diagram than you strictly need to know, and it is so that those of you who are going on to university to do something biological, that it will make more sense when you study respiration there. However, I am going to make clear exactly, those of you who are not going on to study biology at uni, what you need to know in terms of exam answers for A-level. So hopefully it makes a bit of sense that if we're going from a 6-carbon compound to a 5-carbon compound, then we're losing a carbon dioxide. If you wanted to use the very correct terminology, this is called decarboxylation. Not a term you have to use at A-level, but for those of you going on to uni, might be useful. Decarboxylation kind of logically means taking off a carbon. And in the Krebs cycle, it happens twice, again, reasonably logically going from a 5C to a 4C. Now, each of these processes, for those of you going on to uni, can be called oxidative decarboxylation because at each stage, NAD is reduced to NADH. And this is exactly what happens in the Krebs cycle. You have 3 NADH produced and you have 1 FADH2. And in addition to that, you have a single ATP molecule produced per Krebs cycle. And as you can see up here from our little summary diagram, that 2 ATP molecules are produced in the Krebs cycles per molecule of glucose. For those of you who are not going on to do biology at uni, here are the crucial things that you need to know that happen in the Krebs cycle. Notice that you need to know very few numbers. You need to know that ATP is produced. You need to know that NADH and FADH2 is produced, but not the numbers. And you need to know that carbon dioxide is produced. But these are the crucial bits of information for those of you who aren't going on to university to study a biology degree. So now we're getting to the final stage of aerobic respiration. Now, so far, we have got a net production of 4 ATP from glycolysis per glucose molecule, plus another 2 ATP from the 2 Krebs cycles that occur. So we've got the sum total of 6 ATP molecules from 1 molecule of glucose, which is frankly pathetic. Because as we know, aerobic respiration produces lots of ATP. And the actual number of ATP is in the mid-30s. So we clearly have a long way to go. And it all happens at the end of aerobic respiration. It kind of finishes on a high. So now I'm going to draw in the crucial proteins that are embedded all the way along throughout these inner membrane of the mitochondria, along the many cristae, because the cristae increase the surface area for these proteins. But I'm obviously just going to draw one set for simplicity. And these 3 proteins here that are embedded in the inner mitochondrial membrane are the electron transport chain. And this rather unusual shaped molecule is ATP synthase. So what happens? Well, this is where this whole funneling occurs into the electron transport chain. And the thing that is funneled into the electron transport chain, funnily enough, is electrons. But these come from the hydrogen that are being carried by the NADH and the FADH2s. All of the NADH and the FADH2s carry hydrogen so that electrons can be funneled into the electron transport chain. But hopefully it makes sense that for us to get electrons from the hydrogen atoms, we need to split those hydrogen atoms to form hydrogen ions or protons and electrons. So notice I'm not trying to balance anything here. I'm simply trying to show you that the hydrogen atoms being carried by these coenzymes are split to form protons or hydrogen ions and electrons. And these electrons go down the electron transport chain, releasing energy at each stage. And that is why I'm trying to denote here that these bounces, as it were, are getting smaller as they go down the electron transport chain. And the energy released causes hydrogen ions to be pumped across the inner membrane into the intermembrane space. And as a result, we get a build-up of hydrogen ions in this intermembrane space. And that creates an electrochemical gradient. And that is like water being held behind a dam. It's a form of potential energy. And what happens is that these hydrogen ions are then channeled through because they want to diffuse back into the matrix of the mitochondria. And the only way that they can get through the inner membrane of the mitochondria is through ATP synthase. And as they go through ATP synthase, ATP is produced and is produced in very large numbers. So this is how the very large amount of ATP is produced during aerobic respiration. It's at this stage of the process. And this is called oxidative phosphorylation. And this is called oxidative phosphorylation because it is formed as a result of multiple redox reactions as the electron leaves one protein carrier and is gained by the next. Obviously, oxidation is loss, so when the electron leaves, this protein is being oxidized, and as the electron is gained by this protein, it is reduction. And as a result, this is called oxidative phosphorylation. Phosphorylation literally meaning the addition of a phosphate to ADP. This is in contrast to this form of ATP production, which is called substrate level phosphorylation. Both of these terms you need to know, and you need to know where they happen. The third way in which ATP is made is photophosphorylation, which occurs in photosynthesis. So we now need to deal with what happens to the electron at the end of this electron transport chain. And here's another crucial piece of terminology, is that it meets with half an oxygen atom because the oxygen is the final electron acceptor for the electrons coming out of the electron transport chain. And then the oxygen combined with electrons and some hydrogen atoms go to form water. And this is the water being produced here in your GCSE version of respiration. And as a final bit of detail, the oxidative phosphorylation that is happening here, the production of ATP, occurs as a result of a process called chemiosmosis. And chemiosmosis is this buildup of hydrogen ions in the intermembrane space and them being channeled through the ATP synthase. And there we have the whole of respiration on a diagram of a mitochondria showing where it happens and how it happens. It is the whole process, all of the details that you need to know for exam answers. And now we'll go through a few exam questions just so that you can see exactly the terminology that you need to use.
So now we're going to look at some exam questions from a number of exam boards to show you that the information that I have just taught you is going to help you regardless of your exam board. So first question, describe how acetyl coenzyme A is formed in the link reaction. Well, this is as a result of acetyl CoA reacting with the pyruvate and that being oxidized to produce acetyl coenzyme A, with carbon dioxide being released and NAD being reduced to NADH. Describe how ATP is made in the mitochondria. So here the crucial thing is that you remember that ATP is made in two ways in the mitochondria. It's made in the Krebs cycle as a result of the 5C compound being turned into the 4C compound. And it is also made during the electron transport chain as a result of oxidative phosphorylation. And I have just remembered that I should have said, here's a little asterisk in my answer, that it's made in the Krebs cycle as a result of substrate level phosphorylation. So back to oxidative phosphorylation. It's made as a result of electrons going down the electron transport chain, and that causing the pumping of hydrogen ions from the matrix into the intermembrane space. The buildup of hydrogen ions then causes them to diffuse through ATP synthase back into the matrix, which is when ADP plus PI reacts to form ATP. Oh, I didn't even need to say substrate level phosphorylation after all of that. Didn't talk about specifically the production of NADH, so I wouldn't have got that mark. I don't know if I'd have got that mark either. That I would have got, that I would have got, that I would have got, that and that. So I would have got my marks even though I missed out those 2 points. Here we've got a diagram and it's showing stages of the aerobic respiration. So substance X, that is pyruvate, because only pyruvate can be actively transported into the mitochondria, glucose can't. Which stage isn't represented? Well, this is the link reaction, and this here looks like a very simplified version of oxidative phosphorylation, or the electron transport chain. So therefore, it's the Krebs cycle. Explain why oxygen is needed for the production of ATP. Now, here is a classic question where I notice down here that it's 3 marks. And I know immediately that my headline answer, the first thing that comes into my head, is that oxygen is the final electron acceptor in aerobic respiration. And then I'm kind of thinking, well, what else do I write? And so I remember that explain means to describe the biological process and add some more detail to my headline. So I think at that point that I would be talking about the electrons combine with protons and the oxygen to form water. But I haven't, now I've looked back at the question and I haven't addressed this part of the question here. That oxygen is needed for the production of ATP. So they want me to link oxygen with the production of ATP. At which point I'm then inspired to talk about how electrons move down the electron transport chain, and it is as a result of hydrogen ions going through ATP synthase that ATP is made from ADP and PI. So that's right, that's right. Electrons pass down the electron transport chain. Oxygen is a terminal electron acceptor. So even though I went through all of that detail about hydrogen ions, wasn't going to get me any more marks. And it forms water. But notice how I ended up getting those marks from my headline answer and from rereading the question to get inspiration from what is in the question. Describe the production of ATP by substrate level phosphorylation in different stages of respiration. Now there is a clue, there is more than one. And with reference to the number of ATP molecules produced. So here ATP is produced as a result of substrate level phosphorylation in two parts of respiration. Number one, it occurs in glycolysis, and it occurs when triose phosphate is converted into pyruvate. And 4 molecules of ATP are produced during that stage. However, 2 ATP molecules were needed at the start of glycolysis, so the net production of ATP from glycolysis is 2 ATP. The other place where substrate level phosphorylation occurs is during the Krebs cycle, and that is when a 5-carbon compound is converted to a 4-carbon compound as a result of ADP reacting with PI. Okay, so so the reference to that, if I'm really honest, I'm not sure I might have gone into all that detail unless I'd seen this on the mark scheme previously. This I definitely would have said, and I think I would have said at the very least that a net production of 2 ATP molecules are made. And 1 ATP is produced per Krebs cycle. Now that's interesting. I didn't say that. I missed that, even though I absolutely know it. And I did say when a 5-carbon compound is converted to a 4-carbon compound. Outlined processes in the generation of ATP through chemiosmosis. So we're really going to have to concentrate on those hydrogen ions being pumped into the intermembrane space. I think I would go from the hydrogen atoms being carried by the coenzymes NADH and FADH2 being split into protons and electrons. The electrons are passed down the electron transport chain, losing energy at each stage, and that energy is used to pump hydrogen ions from the matrix across the inner membrane into the intermembrane space, where a buildup of hydrogen ions takes place, which results in those hydrogen ions diffusing back into the matrix via ATP synthase. It is ATP synthase that causes ADP plus PI to produce ATP. And I'd probably say, and lots of it. This was an OCR question, and the mark scheme is only including the indicative content. For you to get full marks in OCR, it is important that you detail the stages, the reactants and the products, and a range of processes, and that your writing needs to be well developed, accurate, relevant, and clear. So in terms of the indicative content, I talked about it happening on the membrane and the matrix. I would have certainly talked about hydrogen ions going across the membrane and ATP synthase. That I talked about. Now, I talked about a buildup of hydrogen ions, but then as I talked about the fact that it diffused back down through ATP, that absolutely implies a proton gradient created. I didn't discuss proton motive force, and I didn't talk about this. I talked about it going back through ATP synthase. And I didn't say that. So to be honest, I got 5 out of the 6 indicative marks. I hope this video has helped you. If it has, please tell your friends about my YouTube channel. Please share, please subscribe, and do leave a comment. I really enjoy reading them.